A new kind of cell has just been discovered in the human body. The human body has long been regarded as one of the most intricate and fascinating systems in nature. For centuries, scientists have explored its depths, uncovering mysteries that have led to groundbreaking medical advancements. Yet, despite this extensive research, new discoveries continue to emerge, challenging our understanding of biology and opening up new frontiers in medicine. One such discovery is the identification of a new kind of cell in the human body, an event that has sent ripples through the scientific community. In this comprehensive article, we will delve into the details of this discovery, exploring its implications, the methods used to identify the cell, and the potential impact on medical science. From the cellular structure to its function and role in human health, this newfound cell offers a glimpse into the uncharted territories of our biology. Please subscribe to make 100 subscribers. The journey to discovery. A brief history of cellular biology. The study of cells began in the 17th century when Robert Hooke first observed cork cells under a microscope. Over time, the development of more advanced imaging technologies allowed scientists to uncover the complexity of cells, leading to the identification of various cell types and their functions. From neurons to epithelial cells, each discovery has provided a piece of the puzzle that is the human body. Despite these advancements, the human body remains a biological enigma. With trillions of cells working in harmony, the possibility of undiscovered cell types has always existed. However, identifying these cells requires cutting-edge technology and a keen understanding of biology. The role of advanced imaging techniques. The discovery of this new cell was made possible by the use of advanced imaging techniques, such as single-cell RNA sequencing and high-resolution microscopy. These tools allow researchers to analyze cells at an unprecedented level of detail, examining their genetic expression, structure, and interactions within tissues. Using these technologies, scientists were able to identify a previously unknown cell type that had eluded detection in earlier studies. This breakthrough underscores the importance of technological innovation in scientific discovery. The newly discovered cell. An overview. What is this cell? The newly discovered cell, tentatively named interstitium progenitor cell, IPC, resides in connective tissues throughout the body. It is characterized by unique genetic markers and a distinct morphology that sets it apart from previously identified cell types. Structural characteristics. 1. Size and shape. The IPC is relatively small and has a star-like shape, with thin extensions that connect to surrounding cells and tissues. 2. Genetic markers. Researchers identified a specific set of genes expressed exclusively in IPCs, including markers linked to cellular repair and regeneration. 3. Localization. These cells are predominantly found in connective tissues, including the skin, lungs, and gastrointestinal tract. Functional insights. Preliminary studies suggest that IPCs play a crucial role in tissue repair and immune response. They appear to act as a bridge between the immune system and the structural integrity of tissues, contributing to both healing processes and defense mechanisms. Implications for medical science. Potential role in regenerative medicine. The discovery of IPCs could revolutionize regenerative medicine. Their ability to repair tissues and modulate immune responses makes them a promising target for therapies aimed at conditions such as 1. Wound healing. IPCs could accelerate the healing of chronic wounds and injuries. 2. Organ regeneration. These cells might be harnessed to repair damaged organs, reducing the need for transplants. Impact on autoimmune disorders. Given their role in immune modulation, IPCs could provide insights into the treatment of autoimmune diseases. By understanding how these cells interact with the immune system, researchers may develop therapies that restore balance in conditions like lupus or rheumatoid arthritis. Insights into cancer research. Interestingly, IPCs may also play a role in cancer biology. Their presence in connective tissues and involvement in tissue repair could influence tumor growth and metastasis. Further research may uncover how these cells can be targeted to inhibit cancer progression. The discovery process. Single-cell RNA sequencing. The journey to uncovering IPCs began with single-cell RNA sequencing, a technique that allows researchers to analyze the gene expression of individual cells. By examining thousands of cells, scientists identified a unique genetic signature that did not match any known cell types. Validation through microscopy. High-resolution microscopy was then used to visualize IPCs within tissue samples. Advanced staining techniques highlighted their distinct morphology and interactions with surrounding cells. Functional studies. To understand the role of IPCs, researchers conducted functional assays, observing how these cells responded to injury and immune challenges in laboratory models. These studies provided valuable insights into their regenerative and immunological capabilities. Challenges and future directions. Overcoming initial skepticism. As with any groundbreaking discovery, the identification of IPCs faced initial skepticism. Some scientists questioned whether these cells were truly unique or merely a variant of a known cell type. Rigorous validation and peer-reviewed studies were essential in establishing their novelty. Unanswered questions. While much has been learned about IPCs, many questions remain. 1. How are IPCs regulated? 1. Understanding the signals that activate or inhibit these cells is crucial. 2. What is their lifespan? 2. Do IPCs persist throughout a person's life, or are they replenished over time? 3. Are there subtypes? 3. Further research may reveal variations within this cell type, each with specialized functions. Ethical considerations. As researchers explore the potential of IPCs in medical applications, ethical considerations must be addressed. For instance, manipulating these cells for therapeutic purposes must be done with care to avoid unintended consequences. Broader implications for biology. The discovery of IPCs highlights the vast complexity of the human body and serves as a reminder that there is still much to learn. It also underscores the importance of interdisciplinary collaboration, as advancements in technology, biology, and medicine converge to unveil new insights. This finding may inspire a new wave of research aimed at uncovering other unknown cell types and understanding their roles in health and disease. It also reinforces the need for continued investment in basic scientific research, which often serves as the foundation for transformative discoveries. Conclusion. The identification of the interstitium progenitor cell is a landmark achievement in cellular biology, with far-reaching implications for medicine and science. As researchers continue to unravel the mysteries of this newfound cell, its potential to advance our understanding of human health and disease becomes increasingly apparent. This discovery serves as a testament to the power of curiosity, innovation, and collaboration in science. It reminds us that even in the well-studied realm of the human body, there are still secrets waiting to be uncovered, secrets that have the power to change the world. Subscribe to make 100 subscribers.